My research is about antifungal activities and the phytochemical profile of leaf extract from variously extracted of Laurus nobilis leaves against selected pathogen fungi. Timeline. The problem with fungi is that they are naturally ubiquitous, requiring simple aspects of growth like water, air, and nutrients. Because of its simple growth, this means that it easily affects surrounding infected many agricultural crops, animals, and thus affected the economy. Scientists are looking for a new compound that can reintroduce sensitivity to fungi pathogenic to plant and pathogenic to human, as well as be easy to manufacture and produce, and allow their application safely without here being harmful side effects on the environment or living creatures. Therefore, our research focuses on one of the plants, which is our Laurus nobelius, the leaves one, which are colored by leaves. This plant has been proven by many studies to be an anti-cancer, antibacterial, and wound healing activity, antifungal, anti-inflammatory, and antioxidant. The aim of research is return to nature and eliminate a major environment problem, which is the presence of carcinogenic pesticides in the soil that affect human health and are transmitted through the food chain. Control of agricultural pests that show resistance. Materials and method: first, dried leaves of Laurus nobelius. Second, 6 strains of plant pathogen fungi, which are Cavibarius bisafaria, Fusarium verticillidis, Microphominia fasciolina, Fusarium proliferatum, Fusarium solina, Fusarium uniform. Due to the importance of the crops they infect, in fact, we have chosen these fungi to be economically viable, just as Fusarium verticillidis is often attacks corn crops and to produce a fungal toxin called fumacins that is toxic to humans and animals if they are eating food contaminated with this fungi. We prepared the plant puree, then it was placed in a two solvent, water and ethanol, for 24 hours in a shaker. Then it was filtered and then extracted and mixed with potato dextrose agar medium to obtain different fine concentration 2.5, 7.5, 15, and 30. On the basis of a dry weight of a dried aqueous extract, the disk diffusion method was made to study the morphological and structure change. Second, we use three communal antifungal, one of them radiomyl gold, the second one radiomyl, and the third one uniform, and poured with potato dextrose agar medium. The disk diffusion was made and left it in incubator for 7 days. The results, as we can see here, the inhibition percentage of growth, and these are the concentration. We use it in ethanol extract and water extract, and we see the change in the color shape and the color, and that the fungus is trying to grow up to stay alive. The second one, Fusarium verticillidis, Microphominia fasciolina, Fusarium solina, Fusarium proliferatum, and Fusarium uniform. As we can see here, the effect extract becomes even more 100% inhibition of growth here in ethanol, and we see also here and see in the water in concentration 7.5%, and this is a good result. We took a part of the fungi that wasn't seen under a light microscope, and these are the results that come out. As we can see here, compared to the control, a large difference in the size of hyphae and some of them absent of spores. Cavibarius bisafaria, absence of spores in ethanol and water. Also Microphominia fasciolina, absence of spores in ethanol and water compared to the control. To determine the effect of a plant extract on the morphological and ultrastructure A part of microphonia fungus was taken and examined under two types of electron microscope. Second one, scan electron microscope. Fungi underwent structure change, degradation, and fragmentation, spores formation, and much more. Plasma membrane permeability and destabilization. This is a control. The fungi have is intact, containing all the organs. And in ethanol and water, we see change, deformation, and diffusion in all the organs. And the Bacnida, here is the Bacnida, is intact and full of a protein and a nutrient. But here we see clear change and loss of some nutrient in water and ethanol. And in a transmission electron microscope, we picked a fungus, Microphylla fasciolina, and noted the deformates and the change in hypha and Bacnida. This is the Bacnida in control, and this is in ethanol, and this is the hypha. We see change in the size and a change in the shape. We used the antifungal agent to compare them with the result that appeared in a 6 fungi that were exposed to the plant, and it's become clear to us that the inhibition rate was very similar. This is the first one, radium gold. As we can here see, these are the inhibition percentage of growth. In the Fusarium miniform, we see a 0% of growth. The second one, radium, also here 0, and the uniform, also here 0 in the Fusarium uniform. We do to know the active component that affect and lead to this appearance and structural morphological change in the fungus. We do FTR and GC mass tests. FTR, a prepared sample. The water extract from Laurus nobilis was subject on the FTR analysis. And in GC mass, we use the ethanol extract from Laurus nobilis. The results of GC mass, some of them belong to the phenol compound and the others belong to the carbohydrate. Do you watch the fungus reduce its ability to produce spores and the ability to divide and the loss of organs, and also in FTR we find the component alcohol and alkaline. We get that the extract of natural materials are great and excellent source for using in biological control of fungi. It is safe because it doesn't interfere or cause toxicity to humans, because the toxicity is transmitted through the human food chain. So it's considered a safe and environmentally friendly source, and we can use it as alternative to chemical pesticides. Thank you for listening.